Okay, hi everybody. We have finished plant evolution and diversity, and so now it's time to move on to the animals. I'm going to spend this video kind of going over this big question here. How are animals classified? In class, we will discuss the actual groups. All of these groups, remember that you'll be held responsible for knowing them on your diversity notebook. Okay, so let's go. So how are animals classified? The first thing I think that we ought to do is define the term. Animals are heterotrophic. That means that they have to eat. They are multicellular, eukaryotic. Unlike plants, they lack cell walls. Remember, they have cell membranes, but not cell walls. Their bodies are held together by structural proteins such as collagen, and they have nervous and muscle tissue. Okay, so these are the properties of an animal. Classification of animals can be based on a lot of things, and some of these in this list have gone out of favor because of preference for molecular techniques. But since it is so much easier to look at morphological attributes, I want to mention these. I think they're still valuable tools, and I think we can make some sense out of them in terms of their ecological or evolutionary advantage. So the first one is symmetry. There's two basic types of symmetry, radial and bilateral. The second one is tissue layers in the formation of a body cavity. The term for body cavity is a coelom. That's how that word is pronounced here. The fate of the blastopore, so during gastrulation, based on that, we'll go over that in a second. A new characteristic that really wasn't talked about when I took this course was the type of larva. And then there are some others that we'll talk about more specifically within each group. So let's go back to this idea of the formation of the blastopore. And in order to understand that, we have to just very quickly recall how embryonic development takes place. When the sperm fertilizes the egg, the zygote is formed. So this is a diploid organism, and the cells begin to divide. Eventually, they form this hollow ball called the blastula. This ball sort of invaginates. So imagine like sticking your finger in a water balloon. The blastopore basically starts to turn in on itself, and eventually, and this blastopore in some groups end up forming the mouth, and then the anus is secondarily formed when the gastrulation is completed, and in other groups it forms the anus, and the mouth is secondarily formed when gastrulation is complete. Another thing is that this is when cells kind of start to specialize. So you have different layers of cells, the endoderm and the ectoderm here. And in the middle here you have this cavity, and at this point it's called the blastocele. This eventually ends up forming into the digestive tract. So that's one basis for classification is how that happens, and we'll get back there in a second. In the Museum of the Earth trip, one of the questions that was particularly confusing for you was kind of what facilitated this huge adaptive radiation of animals during the Cambrian period. One of the ideas here is the evolution of predator-prey relationships and just more complex ecological relationships in general. But the other, or an other, important facilitator of this diversity that sprouted up so quickly in the Cambrian period was the evolution of Hox genes. And we don't have time to go into this in great detail, but Hox genes are only present in animals. It's a type of gene, it's a family of gene that can produce this huge diversity of animal morphologies. They are mainly sort of in control of what happens along the anterior posterior axis, and they help do things like form limbs or wings or mouth parts or antennae. And just a small change in these Hox genes can actually result in huge changes in morphology. So when you see like fossils from the Burgess Shale with all of these sort of evolutionary experiments, that is made possible basically by this very powerful type of gene called Hox genes. Okay, symmetry is something that is pretty simple to understand, and the outcomes of different types of symmetry, or the implications rather of different types of symmetry, are important and frankly like pretty amazing. In general, you can have an asymmetrical organism like the porphyrians, the sponges, but the rest of the animals that we're talking about have some sort of symmetry. Some are radial, and some are bilateral. So let me show you what this means. Here's an example of a radially symmetrical animal. This is a cnidarian. It's in the phylum Cnidaria. It's a sea anemone. These are related to jellyfish and corals and hydras and things like that. But what you can see here is that there's no front and back. There are multiple planes of symmetry, just like in this planter. Okay, so that's what radially symmetrical organisms are all about. Multiple planes of symmetry, no distinct top and bottom or front and back. In contrast, bilaterally symmetrical animals only have one plane of symmetry. And we're talking about external morphology here. Internally, like we are radially symmetrical animals. Externally. Internally, of course, we're not. We have, only one heart, and one of our lungs is slightly larger than the other, and we only have one liver, and so on. But externally, we are bilaterally symmetrical. And so one of the things that I would like you to do is to think about what the advantage of bilateral symmetry is. Tissues in the body cavity. So we talked about how gastrulation works and the formation of the endoderm and the ectoderm and the body cavity. There's a little more to it than that. During this development, during the blastula stage, and more importantly during the gastrula stage, three germ layers, three tissue layers in many of the animals that we're talking about anyway, give rise to the tissues and organisms. Ectoderm is the outer layer, endoderm is the inner layer, and diploblastic, so diploblastic means two cell layers. Diploblastic animals only have these two cell layers. The animals that we're talking about that are diploblastic are the cnidarians. Okay, so jellyfish only has the ectoderm and the endoderm. Triploblastic animals, like us and like most higher animals, also have a middle layer called the mesoderm. And most triploblasts, as a result, have a body cavity, have an open space between their archenteron and the outside of their body. Okay, so just to recall, this is kind of how it works. So we get this blastopore sort of invaginating in on itself during gastrulation, and we get the formation of endoderm here and ectoderm here. So we start to see specialization in these cell layers, and we'll see this in a little bit when we talk about protostomes versus deuterostomes, that there's two different ways that the mesoderm layer forms in here. Okay, so back to this, let's talk about the three different scenarios that are important to consider when you look at animals and the formation of, or the absence of, the body cavity. The most primitive triploblastic animal is the is a flatworm in the phylum Platyhelminthes. And what you can see here is we do have these three tissue layers, the ectoderm, the endoderm, and the mesoderm, but there is no body cavity here. These animals are said to be acoelomates. Okay, remember, coelom means body cavity, and when you put an A in front of something like that, you mean the absence of it. So these are acoelomates. They have no body cavity. They're solid. In contrast, members of the phylum Nematoda are said to be pseudocoelomates. Okay, they're triploblasts just like Platyhelminthes, but in the pseudocoelomates, we have the ectoderm and the endoderm, and we have this muscle layer from the mesoderm, and this empty space, this fluid-filled space, it's not empty, is this fluid-filled space called the pseudocoelom. And the reason it's a false coelom, that's what pseudo means, a false body cavity, is that it is not lined on both sides by the mesoderm. Okay, so if you look at a true coelomate, sometimes these will be called eucoelomates. And here we have a member of the phylum Annelida. This is an earthworm. We have, again, the ectoderm and the endoderm and the mesoderm. But you'll notice here that the body cavity is lined on both sides by mesoderm tissues. So that's the difference. Now, this used to be a trait that was paid a little more attention to, but we have since discovered that it doesn't generally result in true monophyletic clades, but we will look at this in lab nonetheless, because it's something that's relatively easy to see. Okay. All right, so finally we have covered the fate of the blastopore in various contexts now. We have discussed a little bit about how gastrulation takes place. We've talked about cell layer specialization, diploblast versus triploblast. I just want to elaborate on a little bit of this. Some of these ideas are still sort of conserved in current classification, and so I just want to explain, the differences between protostome versus deuterostome development. The first major difference is at the 8-cell stage. The protostomes have, the cell lines have basically been determined. In other words, the fate of each cell as either endoderm, ectoderm, or mesoderm is already determined, and that's not the same in deuterostomes. At the 8-cell stage, deuterostomes the cell lines are still indeterminate. The other difference is how the coelom is formed, which really means, it has to do with how the mesoderm is formed. You can see that in protostomes, the coelom is formed from solid masses of mesoderm splitting. It's a little bit different in the deuterostomes, where the coelom forms from folds of the archenteron. And this doesn't have a real selective advantage or anything, but it seems to be highly conserved, and so that's why we're talking about this. In some ways, it's actually more interesting that it doesn't really have a selective advantage, because that just means that it doesn't change over evolutionary time. And finally, and I've already mentioned this, but in protostomes, the mouth develops from the blastopore. In deuterostomes, the anus develops from the blastopore. So again, those aren't really selectively important differences, but because of that, they tend to be conserved in evolutionary time.